On September 27, 2002, James Rothman was awarded the Albert Lasker Award for Basic Medical Research for discoveries revealing the universal machinery that orchestrates the budding and fusion of membrane vesicles. James Rothman is interviewed by Lasker laureate and Nobelist Gunther Blobel of Rockefeller University.
My name is Gunther Blobel. I'm working at Rockefeller University, and it's a great pleasure to interview, which I've never done before, Jim Rothman, who just won the Lasker Award and who works across the street from me at the Sloan Kettering. And we have been good friends for many years, and it's a very great pleasure, Jim, to interview you. Let me start out and ask you a question more in a historical context. How did you decide to work on what became your main theme during your entire scientific career? How did you have the courage to go after that very difficult theme?
Oh, no, that's interesting. If I analyze what it was that took courage, I probably didn't think that it took courage at the time. So what I was really intrigued by was, as you know, Gunther, the secretory pathway. And I learned about it for the first time when I was a medical student. And actually, that's where I really became quite intrigued, in a histology course, in about probably 1971 or so. And it was overwhelming to me, really. George Pilate had just done his really classic work showing that there are transport vesicles and delineating that there is a pathway.
Have you ever heard a lecture of him?
Not at the time.
Not at the time.
But I did later, and that was a wonderful moment. But and of course you were an associate of George's, even a student, so you had that privilege many times. But I didn't. But what I did hear is Betty Hay, I believe it was, giving a lecture to the Harvard medical students. And what was really amazing to me was to hear that there were these vesicles that were budding off of the endoplasmic reticulum and fusing with the Golgi and going from the Golgi to the cell surface, and that the cell was so wonderfully organized, and I really couldn't even imagine how all of that could work. And that's really what captured my attention.
Yeah, I mean, capturing your attention is one thing, but then go and actually decide to work on it and to develop essays is another thing. So how did you?
Okay.
How is the pathway from the initial excitement to actually going and deciding to work on that rather than on other exciting things which are also
well? Actually, I have
too.
to say, Gunther, in that almost at that moment, I decided to work on it.
Oh, that's
but I just didn't do it for a very long time.
So it was a coup de foudre, so to speak.
Yes, I mean that was it for me. I mean it was a problem that I never really could let go of, and I didn't have the tools to work on it. I wouldn't have even known how to begin, and that came later. So
you were a postdoc at Harvey's lab at the time. When you, or was it still when you were in medical school?
Yes, I was just 20 years old, 21 years old, and a medical student. And at that point, I, decided that biochemistry was the way to go. I, as you know, went to graduate school, left medical school at Harvard in biochemistry, and was always working on membrane-related problems. But in the back of my mind, I always had this problem, and I kind of waited, as it were, for the right moment.
And the right moment came when you went to Stanford.
Yeah, pretty much. And the reason...
And that was in
1978.
78,
yes.
Yeah. And so you were on your own, you were a young assistant professor, you already had some beautiful papers on lipid flipping, on how membrane proteins get into the membrane. There's where we overlapped a little bit.
Yes, that's right.
And then you went to Stanford with these credentials, and you now started your own lab, and you said, I'm going to start it with a brand new approach. And
right.
Am I correct?
That's absolutely right.
Thinking?
Yeah.
And so, and even if I'm not going to promote it at Stanford because it could not have worked, and what would you have done? So it was a tremendous risk if you look at it from now, from our.
Yeah, it does. I have to admit, with the. In the hindsight of middle age, it absolutely seems like like a risk. At the time, when
it was an adolescent.
It didn't seem like any risk at all. It seemed like it was something that had to be done. It was the next logical step. And I have to admit, after Eric Fries and I tried for over a year and failed, it began to dawn on me that it might not work.
So maybe you can give us a little bit of a how you actually did it, and what the first experiments were, and what systems you used, and what mutants you exploited, and why it became an assay that convinced the scientific community the minute it was published that it is another fantastic assay to look at very complex reactions in cell biology.
Well, the assay that we developed came in stages, and in the initial stage, what we did was to make homogenates from two lines of fibroblasts, one of which has the so-called VSV-G protein sitting in its membrane due to viral infection. And this is a protein that is normally transported in the cell from the endoplasmic reticulum following your pathway into the endoplasmic reticulum and the Pilati pathway out of the endoplasmic reticulum through the Golgi. And we used a mutant line of cells that is missing a particular sugar transferase. And one of the reasons why this step towards reconstitution couldn't have been done up until about that point, why it wasn't a ready problem earlier, is simply that there were no mutants available that were defective in the glycosylation pathway in the Golgi. And so that was a critical ingredient. So we had one population of cells whose membranes would harbor the so-called VSV-G protein, a system I learned about actually, and you mentioned Harvey Lodich, in Harvey Lodich's lab as a postdoc. And we would break open those cells, Eric Fries, my first postdoc, and I, who did this together. And then a second population of cells that was not virus infected, therefore did not have the VSV-G protein in its membranes, but it was a wild-type cell, and therefore had a good copy of the sugar transferase in the Golgi for N-acetylglucosamine, which was mutated in the mutant. And then the experiment in its initial form consisted of mixing those two homogenates, and what we observed was that the G protein, which originated in the membranes from what we called then the donor cells, became glycosylated by the Golgi, by the good copy of the sugar transferase present in the acceptor cells. And that was what provided the first evidence for movement in a test tube system between membrane-bound compartments, in this case in the context of the Golgi. We learned more about the assay over the ensuing years, of course. But initially,
the key thing is that you could assay for glycosylation by using this mutant, and the other key thing was the donor and the acceptor cell.
And the reason is that, in fact, I have to say the reason why many people felt that this was a surprising thing that you could reconstitute transport in an extract. I'm sure you'll remember that field was very strongly driven by anatomical considerations. One of the remarkable things about the secretory pathway is its very precise anatomy, so beautifully found by George and his colleagues at the electron microscope level. I mean, you have the endoplasmic reticulum facing the Golgi in certain ways. Vesicles that are involved in transport are physically in between the two in the cells. So it was very surprising to many cell biologists that you could break up the cell, destroy this pre-existing organization, this very beautiful organization, and yet the what this experiment demonstrated is an inherent biochemical specificity that must exist to govern the process, which is what kept us occupied for 15 or 20 years and still, in fact, keeps
I
many
guess
people
that's the reason
occupied.
why many people stayed away of attempting that, because there was at that point the dogma that if you mix things up, there may be other structural elements which keep them in close proximity. If you mix them up, and you will never be able to reconstitute. And that's why everybody was very surprised and also skeptical.
Yeah, there was a good bit of skepticism, and that's gone
which
away.
you had to overcome.
Absolutely, and it's gone away in stages over a number of years. And I think really the connection to yeast genetics was very important in that.
But what was the most surprising result? I mean, you found that it was NEM sensitive, this process. You found that you needed energy. Did you expect that is sort of the energy requirement?
Well, I mean, that's to be expected,
The NEM sensitivity, well, that one would not necessarily expect. But did you expect something else? Did you? Of course, you showed minus supernatant, plus supernatant, and you did some fractionation on this. And but everything, the outcome you more or less expected, or?
Well, that's interesting. I think what really is the most was the most surprising for me at the time was the absolute generality of the molecular machinery, and the beautiful simplicity that was revealed when it was found. And so, over the years, as we developed a deeper and deeper understanding, we learned how a vesicle is sculpted out of a membrane by a coat, and the coat is such a beautiful thing because it reminds you of a kind of Buckminster Fuller dome.
But you're already ahead of what I wanted to ask you next. So now you had a cell-free system, and you knew that energy was required, and you knew that the transmit was NEM sensitive. So there was an NEM sensitive component. So the next step is obviously to subfractionate the system and isolate components. So, and can you tell us how you did this and what your first component was?
Well, the first component was a protein we called NSF, which stands for N-ethyl malamide sensitive factor, and we learned quickly enough it was involved in fusion, so we renamed it N-ethyl malamide sensitive fusion protein. Fortunately, the abbreviation was the same, so there was no embarrassment. And what we observed was that when the transport reaction was inhibited with the sulfhydryl reagent NEM, there was an accumulation of vesicles that could be seen, and apparently the fusion process was interrupted. And we were able to purify from cytosolic extracts the protein NSF that was responsible. So that was the very first protein. And in some ways it was the most important protein because by the late 1980s we were able to clone and sequence the gene for NSF. And when we did, lo and behold, it was closely homologous to a protein that was identified through genetic means in Randy Schekman's work called Sec18. And at the time it wasn't known that Sec18 was particularly a fusion protein, but it was very clear it was required in a living yeast cell. And that observation really was, you mentioned earlier the fact that there are always skeptics.
Yeah,
and
that was the first
so.
link.
It really linked them
Randy's
together.
Genetic approaches
yes,
and your cell-free approaches,
yes.
and it really vindicated your cell-free approach. And of course,
Absolutely.
it allowed you to look at mechanisms. So the NEM-sensitive factor, NSF, NEF, was the first component. And then, of course, we all remember the brilliant series of experiments that followed from there. And every 2 or 3 years, there was this wonderful peak and this wonderful new, yet another component that is involved in this pathway. And so what was the next component that you isolated?
So starting from NSF, I mean, the work really followed 2 lines. One line stemmed from NSF, and the other stemmed from this energy requirement for GTP that you alluded to earlier. Following the NSF line, we knew NSF was required for fusion. It was a water-soluble protein. And so we wondered how it worked. And we asked the question, does it bind to the membranes, to the Golgi membranes, which you might think it would do if it's involved in fusion? And the answer is it does, but it required yet another factor from the cytosol in order to bind. And that factor we called SNAP, soluble NSF attachment protein. And we then were able to purify that factor, SNAP protein. So now we had 2 pure proteins, NSF and SNAP. So that was number 1, that was number 2. And we found next that the SNAP protein would bind to Golgi membranes by itself, even without NSF. So there must have been a receptor for the SNAP protein, and we call that the SNAP receptor, which is abbreviated SNARE, or SNARE, which turned out to be a very good name, again, for unpredictable reasons. And so the next step was to purify the SNARE proteins, which could be done by virtue of their affinity to the SNAP protein, which we had in hand as a recombinant protein, and the combined affinity to NSF. So at that point, and I'm bringing you up to, we've now moved from, science is a really long-term enterprise. We've moved from when the reconstitution was first done, 1979, published 1980, up to about 1992, 1993. And at that point, Thomas Solner joined my laboratory at Sloan Kettering and undertook the challenge of trying to purify the SNARE proteins, the SNAP receptors. And you asked me what was a real surprise. I would have to say that that was the greatest surprise that I've experienced so far in science, which is the finding of what those proteins really were.
Well, one of the remarkable findings, and for an observer, an outside observer, always astonishing, was with what speed and what elegance you moved from one system to another. I remember you started out with tissue culture cells, with the mutants, and then you took another biological material, namely brain. You isolated many things from brain, and so and that was another very important thing, because it established the generality of these things for essentially any tissue and any vesicular transport system. And but when is my interpretation correct that you went to brain because there's plenty of it, and what was the rationale for doing that?
I think what you're alluding to, Gunter, is that the snare proteins were first isolated from brain.
Right.
And as a result of that, we learned what the basis of release of neurotransmission really is. Right. And that was not planned. That was absolutely not planned. I wish I could say that it was. It would have been really wonderful to have planned it. But we used brain because we surveyed a number of tissues, and we found quite simply it had the highest level of SNAP receptors. And as you said, there was a lot of it. So that's just what we did.
How did you assay for that? You did this by ligand binding clots?
Yeah, by binding SNAP protein. So the only thing we knew about the SNAP receptor or snare was that it was a SNAP receptor.
So you ran out brain homogenates?
Brain homogenates, liver homogenates, CHO cell homogenates.
You blotted with SNAP?
Everything but potato homogenates.
I see. And so that you found that brain was
it was the highest. Now, in fact, in retrospect,
we were a little
lucky or not. I mean, the reason why was it the highest? It's the highest because between our two ears, we have a very high concentration of synaptic vesicles storing neurotransmitters, and so it's no accident, of course, that it was so high. And following the route that
but it was also lucky in a way, because some of the proteins that you found had already been identified without knowing what their function
Absolutely
is.
Right.
By cloning and sequencing proteins of the synaptic vesicles, synaptic apparatus. And then, the beautiful thing that stunned everybody is that here was the connection to a huge field in neurobiology, probably the most active field in neurobiology, but how synapses fuse with terminal membranes, and how this process works. And you could connect it at this point.
Well, that was the great surprise. I mean, that to me was it was a wonderful moment to have a universal mechanism, really, that would explain the process of membrane fusion, this very general process that results in so much of important physiology, the release of we've already mentioned the release of neurotransmitters at synapses in the brain, the release of insulin from the pancreas after a meal, and the like. There's so many important areas of physiology, and the cell types that are involved are so different. The physiology really is so different. To find that there's one common mechanism underlying all of that is really it's just astonishing, and that is the wonderful thing about science at a fundamental level, as you well know, because you've had the experience too.
Were neurobiologists skeptical of your finding?
Of course.
And how did you persuade them eventually?
Well, hopefully, I'm not sure that I have.
No, you have. I think you have. But
no, I think the neurobiologists are very interesting scientists in the sense that they are absolutely convinced as a group that the brain is very special, and they are right. But the whole history of neuroscience has been increasing connectivity to molecular biology, cell biology. Right. I think Eric Kandel's work really exemplifies those sort of connections. And so I think neurobiologists were surprised, but they weren't completely surprised. And what I think has what sort of has made the connection of snare proteins as the basis of fusion in the case of the synapse a little harder to accept at first, though it is now well accepted, is simply that there's so much regulation in the case of the synapse. I mean, membrane fusion is always regulated. We don't want to release our insulin. We don't want to grow the plasma membrane when the body doesn't need it. It would be, in fact, counterproductive. But the brain has got an enormous regulatory burden because it needs to release neurotransmitters at precisely timed intervals on scales of milliseconds or even fractions of milliseconds. And all of that regulatory machinery superimposed upon the snares, the core machinery of fusion, adds levels of complexity in terms of experimentation that aren't there in more stripped-down systems.
This became then clearer subsequently.
Absolutely.
But, we started out in 1978, right, with your reconstitution.
Right. We're now up to 93.
And we are now up to 93, so we are talking about 15 years with discoveries coming at a rate of, major discoveries coming at a rate of 3 or 4 years. And that's what you would expect in a complex system like this. Did you feel that this was the end of the tunnel in your essay or in unraveling this, or were there further culmination points? I mean, how about when you did these experiments with vesicle and the V and T snares and specific fusion of those? I mean, that I consider a very, very important experiment.
Well, I think as one digs deeper and deeper in science, if you're on a pathway towards truth, it always becomes more beautiful. And at the end, it usually turns out to be very elegantly simple. But nature does not reveal itself in a linear way. And so you go in fits and starts. And each of these sort of cycles of discovery, in a way, are I think they really exemplify that. So at the beginning of the snare story in the early 1990s, we knew of the snare complex, where the two partners, the proteins forming the complex, are membrane proteins emanating, one from a vesicle, the other from a target membrane. In fact, there's 3 proteins in the initial complex we found, and in other versions elsewhere in the cell there are typically 4. So there are multiple proteins that come together to form the snare complex. The fact that this complex associated with NSF and SNAP made it very clear that it was an important basis for membrane fusion generally. But it would have taken more hubris than I could have mustered to have proposed. I mean, we proposed that it was very generally important. We proposed that there were counterparts to the so-called V snare and T snare in the vesicle and target membrane throughout the cell, and that they would partner in a specific way. That already was a proposal that took a lot of hubris, the so-called snare hypothesis, although it seemed logical enough at the time, and that's why we proposed it. But I think even in my most intellectually aggressive moments, I would not have believed it could be so simple that the snare proteins would actually be the principle of fusion as well as its specificity.
The core machinery.
Yes, but in fact it turned out to be so. And when that emerged, and now I'm up to 1998, when we found that isolated V and T snare proteins put into artificial lipid bilayers would lead to the fusion of these bilayers that really directly establish that they are the principle, the active factor in fusion. They really do the job. That was actually another layer of unexpected simplicity. And then, I must say, a very nice step occurred that you alluded to before, when we were able, because we had purified the snare proteins and we were able to reconstitute them and test their fusion in an isolated system, that opened up another round of discovery, which allowed us to actually go back to test the original snare hypothesis in a really definitive way. There was a lot of evidence accumulating in favor of it, but none of it was absolutely nail in the coffin persuasive. The most direct test, that the pairing of individual V and T snares, which are provided, different family members being provided to different membranes in the cell, that their pairing would dictate the specificity of membrane fusion. The most direct test of that, of course, is to have nothing in the system other than the proteins themselves, and to observe that a patterned fusion occurs, the pattern being a precise or very close to precise mimetic of the flow pattern of proteins in a cell. And that's exactly what we observed.
So these are the experiments with the reconstituted proteoliposomes and the assays that you did to show that there is an exchange of vesicle content.
Right.
And then I also heard of experiments where you express the proteins in vivo in different cell types, and you get fusion there.
Well, and that's actually not even published yet, my friend. But yes, so in very recent experiments, we're finding that we can actually engineer the cells. So we can take the snare proteins and flip them to the outside of the cells using a signal peptide discovered by a certain Günter Blobel. And when we do that, the snare proteins program the cells to fuse, which is a wonderfully vivid demonstration of the generality and the power of this hypothesis.
It is. It is.
Yeah, I mean, we're really excited about that.
And it's surprising that it works on extracellular surfaces as well, because you don't have the other intracellular components that you any NSF and all.
Well, and it really shows though that this is the core principle of the fusion process. And the fact
how efficient is the fusion?
Now we're
anticipation of
now I'm afraid you're going to
publication of
review
this
my article. The answer is very efficient. In fact, so much so that we see cells with multiple nuclei in them, where we're getting multiple rounds. One cell fuses with another, and that wasn't enough for it. It got hungry, and it took another one in. That's very, very efficient. So
an important support for your bold snare hypothesis also came, of course, when the X-ray structure was done, and also when the virus fusion showed that there are relationships in these processes, and maybe you can tell us a few things about.
Well, I'm glad you mentioned that. Reinhard Jahn and Axel Brunger in 1998 published a structure actually after our fusion results. But quite independent, that showed a beautiful sort of hairpin arrangement in which the V snare and the T snare are rods, alpha helices, that form a bundle, a so-called coiled coil, that the structure implies pairs between membranes. And that structure provides a perfect mechanism to explain what we've observed by forming a pin that adheres the two bilayers to each other and undoubtedly takes energy that is made available through the process of this assembly, protein folding, released during this exergonic process in thermodynamic jargon, that would couple to the two bilayers and move them through the transition state for fusion. The structure didn't show that, but it certainly was consistent with that mechanism. So that was a really, I think, a significant
and
event
the virus
in the field.
story?
And the virus story, yes. So quite separately from all the work on intracellular membrane fusion, for many years, people have studied viral envelope fusion, where a viral particle containing a chromosome will enter a cell by either being endocytosed in the cell and then fusing with an endosome, or fusing directly, depending on the virus, at the plasma membrane, therefore introducing its genome. And the active principle in that case is the so-called viral fusion protein, which differs from one virus to the other. These days, the most prominent one is the fusion protein of HIV, and it's the most prominent because it's so central to the initial event of infection that gives rise to AIDS, and also because drugs are now successfully being tested in the clinic that are directed to this. The same type of principle is responsible for infection by influenza virus, a different protein, but as it turns out, the crux, the structural crux of the core of these viral fusion proteins, which was recognized by a number of structural biologists, but most prominently the late Don Wiley and John Scahill, who really put it together in a wonderful review article in 1998 or so. These cores also form pins. They are just like, structurally very similar to snare pins. They're oriented on the other side of the membrane. The sequences of the proteins are different, but you have a beautiful convergence, sort of in physical biochemical terms, an evolutionary convergence, in which what's really important for fusion is the formation of a stable hairpin structure based on so-called coiled coils made of alpha helices that is extraordinarily stable, so much so that current estimates actually suggest it takes 1 billion years for it to dissociate once it's formed, to dissociate spontaneously. And it is that physical principle that allows two membranes to be put together, and it can be utilized on the outside of a cell by a virus, on the inside of a cell by cellular membranes, and there are different sequence solutions that have evolved, but one common principle for membrane fusion. So there's a kind of generality there that's really wonderful.
Yeah, that is wonderful. Now, with a complex pathway that is working in essentially all eukaryotic cells, plant and animals, if we go back to human diseases, there are obviously many human diseases which will be linked to this pathway, and we are only at the tip of this huge iceberg. And maybe you can tell a little bit of the latest developments in that field, relevance to the medical, to the clinic.
I think we've only begun to see the tip of the iceberg in terms of the implications of this for medicine, but the tip has already exposed itself. The disease that first comes to mind, both because of its clinical importance and because of therapeutics that are targeted to this pathway currently in use, is type 2 diabetes, which, as you know, is an insulin-independent diabetes, generally adult onset, and where the principal biochemical problem is hyperglycemia, which occurs despite the fact that, at least early in the disease, adequate amounts of insulin are secreted in response to a meal. And so the muscle especially, but also the fat in patients with type 2 diabetes responds inadequately to the signal from insulin to take up sugar. So the insulin is there, the blood sugar is there from the meal, but the sugar is not being taken up by the bulk tissues, such as muscle and others. And so what's wrong there? Well, what's wrong is that there's a secretory-like pathway, which in fact is the basis for controlling sugar uptake, and it's not balanced correctly. The homeostasis is wrong. And what it is, of course, is that there are transporters for glucose molecules that sit on the outer membrane, in the plasma membrane, but there are also an even larger proportion of these that are known to sit in intracellular membranes. And there's a dynamic equilibrium which is due to rounds of membrane fusion and endocytosis, vesicle budding, the very pathways that we've described. And in what is not happening in type 2 diabetes adequately is this translocation of sugar transporters from the inside endosomal membranes to the plasma membrane. That fusion process is misregulated. The current generation of drugs that are being used for this purpose, so-called insulin sensitizers, are directed at this signaling pathway, and there will undoubtedly be more.
In the synaptic vesicle is recycling, and very much in a way like the glucose transporter.
Yes.
Not completely the same, but,
Right.
but one would expect a lot of diseases in neurobiology. Any
Well, in fact,
insights there already, or any connection there? I mean,
well, yeah, there is. I mean, there's a profound clinical
Yeah.
connection. If you take, for example, mood disorders, or let's say more generally psychiatric disease, with an organic basis, these conditions are treated clinically and with significant success with pharmaceutical agents. The most popular is, best known in the popular literature, of course, is Prozac, that are directed at neurotransmitters. Their pharmacology differs dramatically, whether depending on the chemical compound, but the end result is to modulate the level of a neurotransmitter in a synapse. And so what we know is that clinically successful therapeutics are directed at modulating activity of synapses. Now that pathway by which neurotransmitters and their effective concentration in a synapse is controlled is in fact the very pathway that depends on the snare proteins, both presynaptically through the release of the neurotransmitter. That's a membrane fusion event. There is a more recently discovered membrane fusion event postsynaptically. There are vesicles also in the postsynaptic side of the synapse that have the receptors for the neurotransmitter. And just like the glucose transporter rather, those also undergo a regulated translocation reaction. So I think that as we understand more about the regulation of this pathway, both in the brain and in other physiologic contexts, there will be new targets that will be developed for
I mean, I suppose the secretion
intervention.
of the ligand and the withdrawal of the receptor on the other side.
Yes, it's really the homeostatic balance that
right,
controls the responsiveness of the synapse, and it's
which
that
is done
level
by
of
the same basic mechanisms.
It is.
Yeah.
It is.
So, of course, with many regulatory components, which some of them still need to be discovered. But so, are you thinking about schizophrenia, manic depression.
Well, not at the
some of
moment. With the Lasker Award, I'm not suffering from schizophrenia or Lasker
no,
or
some of the
manic
great
depression,
questions that
yes.
that are not yet solved. And would you be surprised if some of the genes that will be discovered in schizophrenics or manic depressives will have something to do with this pathway?
Your question is really
loaded.
No. I mean, I find it very interesting because it's almost telepathic. Because actually there was a paper published in about 2 years ago that studied prefrontal cortex from schizophrenics versus normals to look to see what genes, if any, were consistently expressed in a differential way. And what they found was that the principal difference between the two was genes that are regulating and in synaptic controlling synaptic activity. And there were actually 2 genes in particular that they pointed out that were very significantly altered in their level of expression. One of them is a protein discovered by Paul Greengard called synapsin, which is known to control the level of synaptic vesicle availability in synapses. The second was the protein we discovered, NSF, which in fact, in a broad sense, also controls vesicle availability and therefore is in the same pathway as synapsin. So there's already evidence of that, and in fact I've just recently started in the laboratory projects that are directed at rapid and efficient testing of large numbers of genes in these and related pathways, in the type 2 diabetes case and also in the case of neurotransmission to identify regulatory genes.
That's where you're going to go in the future, or?
Well, yeah, I mean,
that's one of the directions.
I think for me, well, I think for the field really, the major, I mean, the with the discovery of the core machinery that is responsible for the budding and the fusion of vesicles, we now have some linchpins to rely on to try to understand the physiology. And what we have is a very general core pathway that is used differently in subtle ways, probably some ways that are not so subtle, by different cells and tissues. And they may even be used differently, as you alluded to, in disease situations where there's a chronic imbalance in homeostasis. So really the most uncharted territory is is that of regulation. And you know, I mentioned that earlier in the discussion that I started my scientific career in medicine. I never finished, but in a way this is my return to medicine. And I think it's a responsibility to explore these avenues. You know, I've been very fortunate, as many of us have. Our work has been very generously funded by the government initially, which backed me up when I took a risk, the National Institutes of Health. Now more recently, still from the NIH, but also I've benefited from very generous philanthropy at Memorial Sloan Kettering. The board of Memorial Sloan Kettering has been fantastic in supporting and sharing the vision of science in the service of medicine, particularly cancer, but with a very broad view that includes fundamental cell research. And now is time when one makes some discoveries that can be exploited to pay back. And I'm really interested to do it, and I think it's entirely appropriate.
If you would have another 30 or 40 years of Jim Rothman starting at age 26 or 25, I don't know when you started in science, and you started out with a great vision. And the great vision was that you wanted to unravel the molecular mechanisms underlying vesicle transport, of course not realizing that this would perhaps it would be of such profound importance for how the cell is organized itself. If you would have the luxury to start out very naively again at age 26, what would be your next vision? What would
you mean today?
Yeah, today.
I really want to ask you that question.
I mean, obviously, you have always settled in on a problem, and then you have pursued it, and then you know there aren't that many problems that lend themselves to this sort of analysis.
You mean the sort of reductionist analysis?
Yeah. Have you thought about it that you would, if you would have another 40 years of time and energy and everything that you had when you were 25, what you would do? Let's suppose you would have cloned yourself.
At my age, Gunther, or your age, maybe the thing to do would be to focus on aging. If we had another 40 years. But it's a very serious question because it relates really to choices that young scientists make today.
Yeah.
And one of the great benefits of being a young scientist and making choices that involve risks, as you will remember because you did a similar thing, is that you don't have to take the advice or benefit from the so-called insight of senior scientists. Having said that, if I were to start out all over again, and I had the opportunity to do that now, I'm not sure I would take a reductionist approach quite the way that I did. I would take a reductionist approach, but it would be more at a sort of systems level. I mean, I feel that the major challenge to biology today, and also to medicine, it's really the same challenge. I mean, we have large numbers of pathways, of which the secretory pathway is one, of which the pathway of protein translocation across membranes is another, and there are many pathways that have been beautifully discovered over the last. In fact, that's our contribution to the cathedral of science, our generation, so to speak. Now then, the question is, what does the cathedral look like as a whole? And there is no answer to that. In other words, what we have really, and the Human Genome Project and the completion of the sequence really in some ways focuses on this question. We have 50, 60,000, nobody knows exactly how many genes. They all represent one or other kind of a working part. We've got them organized in sort of linear pathways that make sense for the core activities of a cell that all cells do. But when it comes to physiology that differentiates one type of cell from another, we all appreciate now that there are networks of activity that make integrative decisions, that the cell behaves, and indeed the organism behaves, like a complex computer. A cell has to incorporate an analog environment of factors that say, and come out with a binary decision. A decision to divide, a decision to migrate, a decision to die, maybe a decision to secrete. It's also the result of a network. How those networks really work and how this sort of assembly list of parts kind of comes together in combinatorial ways to provide flexibility, physiology, and most importantly, chronic pathophysiology. The most difficult diseases to address still are those chronic diseases which develop a life of their own beyond their initial pathology, and we don't address them very well, pharmaceutically, sometimes even diagnostically. And all of that relies, in my view, on understanding, developing even a way to understand networks, and the sheer complexity of it is daunting. It may never be solved. Somehow, as a scientist, I don't really believe that, but I do believe that we're going to have to evolve as scientists ways of taking advantage of precise understanding, as you and I have done in the kinds of work that we've done as biochemists, and at the same time a more systems-oriented view, the way traditional physiologists do. I don't know how it's going to be done. I can't even imagine what the solution will look like exactly, except it's critically important. And since it seems like an unapproachable problem, that's the problem that would attract me. It's the single most important problem in biology today. It's such an important problem, it can't even be posed precisely. In fact, when you can pose a problem precisely, when you've reached that stage, then it's an answerable problem, but it's therefore a little bit less interesting for that. So the real challenge, I think, to the young biologists today, if they wish to take it, is to take this sort of very loose description that I've just afforded to the biggest problem of the day and try to find a way, that's where the creativity really is, to reduce it to tractable questions that can be asked and answered.
Yeah.
A long answer to a short question.
We go back to Pallade, who has defined the problems that we were both working on.
Yes, that's
exactly
right.
How do proteins get across the membrane, and then the vesicular transport,
that's right.
both defined by him as phenomena.
Right.
The molecular solution then has worked out. And so there are probably things which have been defined at that level out there that have to be discovered by young scientists who can then, by whatever means, pursue it and make it a long-term project.
That's right.
Which would be very satisfying.
Well, we don't have 40 years, but actually, between the two of us, maybe we do. So maybe we should collaborate on this.
What was your most exhilarating moment in all of these magnificent story that we have heard over the last, let's say, 22 years. Not in terms of rewards or prizes, the Lasker is certainly one of the highlights and high points, but in terms of scientific achievements, what was one of the most exhilarating?
I can tell you the answer to that very precisely, and it comes back to the snare complex. We knew over a period of a couple of months, as Thomas Solner was doing these experiments, when we cracked some basic technical problems, it happened very quickly, over a matter of a month or two. And we were getting sequences, really, of the polypeptides in the complex. They were bands on gels. What were they? What was their identity? And we got two of them, SNAP25 and syntaxin, that came fairly quickly. And we quickly looked, what are they? Okay, well, as you indicated before, they were already cloned proteins. Nobody knew what they did, really. But it was clear that they were major synaptic membrane proteins. And then there was this other band. And Thomas and I were hanging around by the sequencing machine, watching the bases come out one by one. And then I had to go and give before getting the answer, I had to go and give a seminar at Harvard. And I will never forget getting a phone call from Thomas just before, 5-10 minutes before the seminar. And he said, it's VAMP, synaptobrevin. And we both knew immediately what that meant, because it's another membrane protein of the many that are there, but it's one in particular that was in the other membrane, in the synaptic vesicle. And that fact, that was the linchpin observation that meant that we had a protein complex of membrane proteins involving pairing that could bring the membranes together for fusion. And I will never forget that moment of exhilaration, and at the same time the very difficult feeling of concentrating on the lecture that I had to give.
I see.
Which did not include that.
What were the most distracting and disappointing moments? Have you also had moments where you wished you wouldn't have said something, or you wished you wouldn't have published something, or you wished you could take the paper back? That's an embarrassing question. We all had those. Of course. But
Of course. I mean, there have been things that, like most, well, I mean, if you operate on a frontier and in a fog, okay, sometimes you're going to trip on yourself, and it's unavoidable.
Very well phrased.
And so
I will remember that when I ask this question next time.
And so, there will be times when, but what's important, I think, is that at every step that you take in this sort of pea soup is logical, and that at any one moment you make an interpretation that is the best interpretation you can make. So I would say, looking back, there are two things that I thought were that there's one that I truly regret, and it was a bad judgment, but it was an honest experiment, and that is early on. We thought, perhaps, we tested the hypothesis that clathrin-coated vesicles might be the responsible agents for intracellular transport, and we did it by subcellular fractionation. And what we found was evidence but in fact, it's not the correct conclusion, that clathrin-coated vesicles carry VSVG protein into and out of the Golgi. We published a paper to that effect. The experiments, in fact, the result was due to a process we didn't know about, which was endocytosis of the VSVG protein in clathrin-coated vesicles, involved in recycling to the cell surface, and it led to a confusion. The important thing is, I think, that you learn from your mistakes. And what I learned from that is actually listen to other people's advice before you publish, is actually what I learned the most, because I was at Stanford at the time, and Roger Kornberg, my colleague, said, gee, Jim, this evidence comes only from subcellular fractionation, not from any other line of evidence. And what I learned from this was have two kinds of evidence that are independent, that point to a major conclusion, to avoid the possibility of a significant artifact. So that was probably the one flat-out mistake that we made, and I learned a lot from it.
We talked about the 30, 40 years ahead of us. Now, that we know that it's unrealistic, at least in my case, in your case, not. But what are your major plans for the future? I mean, if you have any. that you have made for the future? Which direction are you going
Well,
to
I'm starting
science-wise and also otherwise?
Well, I'm starting actually sort of a new venture for me in genomics, and it's a way of addressing the question of physiology and the way in which our pathway, the pathway of fusion, is physiologically used. And so what we're doing is setting up actually a large-scale genomic project in which we're going to be doing essentially automated microscopy together with, on a large scale, using these recently discovered small interfering RNAs, which are very exciting gene-specific reagents. And Urs Rudeshauser and I at Sloan Kettering are organizing a very exciting program to take advantage of an automated microscope that we've been able to get our hands on, that is a confocal microscope that will do 30,000 essentially cell cultures a day and give us data on a cell-by-cell basis on the activity of the cell based on a readout that we've engineered. And so if we engineer, as we are, readouts, for example, for glucose transport in fat cells or synaptic a neurotransmitter release at synapses, whether it be glutamate, for example, or acetylcholine and the like. And if we can systematically test gene by gene by gene which ones regulate that pathway, we will be able to, we hope, that's the hypothesis, discover the patterns of regulation, and on a large enough scale that we should be able to see what's different for the different pathways, not just what's the same. Because when one wants to intervene pharmaceutically and medically in these arenas where we've alluded to before, you have to know what the targets are, but also what the targets are that are specific to a particular pathway or a particular process. And so this is, in a way, it's my answer to what comes next in the field. For me, this is what comes next. It's regulation. But I'm trying to move the scale up to one that acknowledges the fact that most genes, if not all genes, have already been cloned, try to change the way that we proceed. And at the same time, if there are patterns, network patterns to be revealed, maybe we'll have a chance to have a crack at it. So I guess when you come right down to it, I'm trying to give it another go, even though I don't have another 40 years.
Thank you very much, Jim, for your summary of what you have done and where you're going to go.
Thank you very much, Gunther, for a nice interview. I appreciate it.